All right, and we are live. Welcome to the Willow Creek Cannabis Alliance webinar number 3 on integrated pest management for cannabis cultivation. Don't bug out, people.
Okay, so I already gave you a very brief introduction, but for those who don't know, I am Matthew Gates of Zenthanol Consulting, and I'm here to talk to you about the basics of IPM and a couple of advanced concepts I actually wanted to expose people to very briefly. So I wanted to start off by saying that IPM requires sort of a frame of mind change for some people, and it's best approached in a holistic fashion, by which I mean you consider various aspects of your context, including things like your location, your local area, and your general area outside your cultivation space, which will influence your pest pressure, for example, among many other things. Various environmental forces like your weather patterns and things like that. I already mentioned things like biological forces, but also things like your social forces, the things that are accessible to you, your resources, your regulatory bodies and things like that that will influence what you can and can't use potentially, and various factors like that. All of those things combine to become part of your integrated pest management system, and you have to consider all of these facts and all of these facets. Otherwise, you run the risk of running into a problem or a snag that you can't actually find a solution for. And one thing that I'm very excited about with the cannabis industry in particular is that a lot of people are very keen to sort of coexist with their local environment and act in a more sustainable fashion, which is another important part of IPM, in my opinion. Before I go any further, I wanted to touch a little bit on empiricism and plant health claims. Basically, I can come up here, and the lovely panel that we have can come up here and say things. But I think it's very important that people seek out side sources, subject matter experts, and things like that in their general work with IPM. People will come in, people have very great anecdote and experiential knowledge, but it's also helpful to kind of marry that information with research. And actually, that's one reason why I'm very excited to have Lauren Quakenbush with us here. On the left, we have as an example, a claim about increasing photosynthesis volume to like 600% by, I guess, Don Huber, but I couldn't find anything on it. It's not a very good citation. That's my example. On the right side, we have a research report, what is the maximum efficiency with which photosynthesis can convert solar energy into biomass from 2008. And it shows very clearly, and there's other aspects to it, about exactly how efficient different kinds of photosyntheses are. And it's very, very much lower. So when you see two different claims like that, kind of ask your question, why is it? Why is there such a discrepancy and things like that? And moving forward in IPM, as you find more information and research, you should do that. I also want to introduce Surendra Kedar's IPM cycle, at least that's my name for it. This is the way that research groups look to make outreach about IPM towards people in local communities and also on commercial scales. I think people who are looking to grow cannabis, whether in a private way or in a commercial fashion, should become very aware of the multitude of different aspects, not just the direct pest management things like biological and chemical treatments, but also things like your SOPs, your organizations that support you at various levels of organization. How you communicate and how those organizations communicate with you about what you could or could not do, various best practices, things like that, and also resources. We already have another panelist that I've already mentioned that will probably be able to talk more about that. And also some basics on plant immunity. It's not necessary to have like a super detailed and critical understanding of in-depth physiology of plants to grow really great plants, and indeed cannabis too. But it is helpful to know certain things so that you kind of come away understanding what would maybe make sense and how you can make informed decisions. And I won't go into too much detail here, but basically certain things like the growth immunity trade-off, if plants are getting stressed through abiotic and biotic factors, pests, things like that, then the pathways that link like vegetative growth and bolting and flower development are intrinsically linked to other aspects like how you combat pathogens and insects and things like that. And so it's not possible for an organism like a cannabis plant, for example, to really kind of do both at maximum efficiency. Partly because nothing works at maximum efficiency, but also because even theoretically or practically, stressors and other sorts of things are going to make that difficult and not possible. So you should always be considering what could be stressing out your plants, and also consider that it's not possible for plants to sort of deal with every single possible problem, biological or abiological. Also, symbiosis. People like to throw this word around. Pest, beneficial, mutualist, parasite. At least in the case of the first two, and also the last two, they're human-made constructs. There's no organism that is part of Team Pest or Team Beneficial or anything like this. And I think that's an important sort of frame of mind to come across with, because when you interact with the various organisms like into your cultivation space, they might not even be associated as pests with like cannabis, for example, but you might freak out and think, oh, I have to spray, I have to apply some sort of treatment, when you don't actually need to. And that saves you time, it saves you energy, it also saves you money, and also saves you some stress yourself. And also things like mutualisms and parasitisms, these are a spectrum, and anyone who's interested can look further into some of the research reports that I cite at the bottom, all these titles at the bottom here with the dates. You can look into that, but basically, ecological theory tells us that various organisms can be pests in one context and beneficial in another, or they can be mutualists in one case and parasites in another. And so I think that's also a thing to really consider. This is nuanced and complicated stuff sometimes, and it's very rarely simply a basic answer for all cases. There's also this thing called the hologenome theory of evolution, which I think many people in agriculture can appreciate. It's the idea that, or at least it's one idea that sort of explains how we are all sort of connected, our microbiomes, our genes, the things that we associate with. And without going too much into detail, I think people can understand that the cannabis plants that we grow are sort of similar. There's their own genes, there's those own microbes that live inside them, on them, and around them. But there's also organisms that we input, that we associate with our crop, ourselves included. And we can also be vectors for pests and disease and things like that. And so can things like the wind and other insects and other things you come across. So it's all connected. And if you start growing with that mindset, I think that helps you avoid certain pitfalls. Some of those are sort of invisible stressors that we might be aware of, like temperature, humidity, various toxins, heavy metals, things like that. We might be aware that those things can exist or even be currently active in our area, our cultivation context. But we don't always understand exactly how those things kind of sort of work in tandem to be a complete and net stress force. At any one time, a plant can be dealing with various facets of its metabolism. How is it going to deal with oxidative stress, like all organisms do, various pathogens, and in our case, like insects and mice that feed on it. Also things like wounding damage, like a branch falls off, or a hurricane happens, or something extreme like that can happen and wreck a plant. No matter how healthy it is, it can be buffeted and hurt in ways that it will have to recover from. And many people like to talk about the phytomicrobiome, and very briefly, I just want to touch on the fact that the immune system of plants requires a very it associates with various microbes. And sometimes, like I said, those microbes can be beneficial typically, or typically detrimental. Sometimes even the typically beneficial microbes can alter or help out, typically pathogenic or at least not very helpful microbes. And again, it's a spectrum that can change in a short period of time and a very long period of time. And it goes without saying, but like this diagram sort of mentions, the immune system of plants is at its lowest at the end of the life and during senescence, and in the beginning of life, like when it's a seedling. And when it's maturing, that's when it's the most strong. And I think that's very relevant to people in cannabis for a number of reasons, like when you keep mother plants, like when you're trying to sort of, end on a high note with your harvest as your plants start to age and get to that point where you're going to harvest, sometimes the immune system can become a little bit more finicky to deal with. I'm not going to go into too much detail here, but I thought it'd be a great reference to make that various bacteria, fungi, mycota, things like that, they get detected by plants because of their physical structure. They penetrate the plant with penetration plugs, with enzymes, with things like that literally melt and break down and dissolve, in some cases, the plant tissue. And then plants, they detect that, they create they create reactions to that. It creates a local alarm response that also stretches to and fro from the top to the bottom of the plant in some cases. Then the pathogen has sort of a reaction to that. It creates effectors that kind of neutralize that. And then the plant makes countermeasures that neutralize those effectors, so-called effector-triggered immunity. And without going too much into detail, like I said, this kind of turtles all the way down, and just don't make any mistake that the plant immune system or the comprehension of it is a very simple or very basic thing. There's a ton of things that are going on with plants all the time, constantly. Yeah, like I said, here's an example of various pathogens, particularly fungi and bacteria, that can penetrate plants in the rhizosphere and in the phyllosphere, in leaves and in roots. Through, at least in the case of leaves, through stomata even. So stomata open up, and bacteria can go right through into the xylem channels where water comes out. There are flaws to the immune system that are inherent and natural. Sometimes, though, there is no opening, and an organism like a fungus, like a powdery mildew, for example, will create a sort of a prosorium and penetrate through and create a little root kind of a organ, like a hostorium, and siphon the resources out of the cells. It needs the organism, the host, to be alive for it to live. This isn't true for all pathogens, but it's true for some. And in that case, a lot of detrimental or pathogenic microbes actually evolve or are very much closely related to beneficial organisms that use the exact same or similar pathways to interact with the plant, for those who didn't know. And then, immune system signals in plants when these things happen can happen as quickly as 1 minute to 10 minutes to 30 minutes, and in some cases even 24 hours or longer. A lot of people like to utilize systemically acquired resistance and sort of prime the immune system of plants, which I think is an important and overlooked aspect of IPM. But it's important to kind of know how those dynamics function and how elicitation works to, in comparison to when a pathogen actually attacks. These elicitations can be helpful, but again, remember, your plant is pulling resources from somewhere else in order to prime its immune system and utilize its immune response. Similar things happen with arthropods. Here's a great example pulled from Genomics of Adaptation to Host Plants and Herbivorous Insects from 2015. There's myriad ways that insects, just like the microbes I mentioned, will feed on a plant and will use their own sort of either microbiome or endogenous enzymes and toxins and other sorts of things to sequester resources and to sort of degrade the plant immune system. Insects have been doing this for about 400 million years, and they're probably not going to get any worse at it. I do have a thing about mycota, but I really wanted to end there because I know that we have other panelists to talk to. So again, if you'd like to learn more, you can find more of this information on my YouTube channel, Zenthanol. You can also find me on my Instagram channel at SyncAngel, also on Twitter at SyncAngel, and you can find my Patreon, which is Zenthanol, or you can contact me at zenthanol.com. One big problem would be that I often see in the cannabis space, and also traditional agriculture or other agriculture, cannabis is traditional agriculture, is that people will simultaneously utilize they'll add a lot of phosphorus or other sort of nutrients. Specifically, I mentioned phosphorus because then they'll come back and add mycorrhizas. And there's a lot of research that goes over the fact that certain mutualisms don't develop unless there's a need in certain cases, because even mutualistic relationships between a microbe and a plant they have a cost to them. That's why they're delicate, and that's why plants that escape their original area or other organisms that do will they will lose the genes and other factors that are necessary to initialize those relationships. And so, like if you have tons of phosphorus, then mining inorganic phosphorus for mycorrhiza or for plants and trading that for hexose, for sugars from the plant to the mycorrhiza, in certain cases, it doesn't make sense to do that because it's already very much accessible. And so the prerequisites actually aren't there. And there's other cool things like that, those nuances that exist.
Yeah, that's fascinating. I just sort of came in the last year that came into the field of view too, where it's like, if you have plenty of fertilizer, is the plant even developing the relationship with the mycorrhizae because it's already got it all accessible? How I don't think we know enough about that. I think Tad from Kiss Organics had a good podcast with some Israeli guy. I can't remember his name right now, but it was all about mycorrhizae. And they covered it in some of that. That's a great podcast to listen to.
I have read those studies, and it is true that when you have an excess of phosphorus available, that the plants are less likely to form that mycorrhizal relationship with the plants.
And it's like and even like that is just a sliver of an example in a sea of mutualisms and parasitisms out there, right?
Well, and I think ideally too, when you're looking at nutrient deficiencies, that there's a lot that can be done without having to introduce fertilizer. If you're regularly adding compost, if you're regularly adding things like composted manure or biochar, that those things are going to increase your nutrient transfer and increase really organic matter processing within your soil. So yeah, it's not always necessary to add a bunch of fertilizer. However, if you are growing from like fertilizer bags, those typically only have about a 30-day availability of nutrients. And that's why growing in your soil can be so valuable, because you don't have to keep adding nutrients. If you're adding compost once or twice a year and maintaining a healthy amount of organic matter and microorganisms in your soil.
I'm curious to the panel here, what relations in regards to Brix and how can you gauge your plant's Brix readings with quantify its health through that, and how do pests and disease react to fluctuations of Brix levels throughout the plant's life? Can anyone speak to that?
I mean, even in high Brix situations, I still see pests in my experience, and I've seen research that shows that as well. Sometimes even I have a video on my YouTube channel called gosh, I guess I'm forgetting the exact title, but it's about IPM and plant insect digestion, gosh, I can't believe I'm blanking on the title. But anyways, it talks about how specifically with insects, how the digestion and the consumption of plant materials is affected by various aspects of plant physiology, and Brix is one of them. A research report in 1996 that I cite in the video talks about how leafhopper nymph levels, they were actually looking at claims about high Brix levels getting rid of or repelling piercing, sucking mouthpart insects. And what they found was actually the higher the Brix level in these vineyards, organic and conventional, the higher the Brix level, the higher the leafhopper population actually. So that's an example that I think a lot of people might, if they're familiar with high Brix being related to like high plant health or other factors of physiology of plant, that's not actually the case, or at least in some cases. Again, I think plant health is super nuanced and also kind of vague. When people say my plant is healthy, like, what does that really mean? I've read a lot of research about even human health and plant health, and how people have asked the exact question I just asked a lot, and there's not like a really great definition everyone sort of uses. There's no perfect definition, of course. Keeping a good record of what has happened week to week, sometimes even day to day or year to year, is invaluable. I recommend that people, if you're trying to up your IPM game, this is along with crop scouting every week, or as much as you can. I would also include like keeping a record you can access easily, a record that is preferably digitized, and in a commercial setting, a record that has a way to interpret data and turn it into a visual. I think that's super invaluable for people. I've used scouting programs and things like that have shown over time, like, here's where we can see the populations of the pests and the populations of the biocontrol, for example, sort of crest and ebb and flow. I can also see when we released and how many we released, that kind of stuff is really helpful. And I think in the future, we'll also even have access to things like ways to like sequence, plant microbiomes and things like that, potentially, or other sorts of like microbial contacts, so that people who are trying to sort of facilitate certain microbial populations that they find, that they collect, they can be sure that they aren't introducing, sort of something that might not be great, because most microbes actually haven't been cultured. Most bacteria, for example, I'm pretty sure most fungi as well. So, even the stuff we're sure of. There is a company
called Trace Genomics, out of San Francisco, that is beginning to create a soil profile of soil microorganisms. And like any genetic database, its wealth is based upon the number of growers that are sending in samples. So if you think that you have some anomalous soil fungi or virus, then please reach out to them and please send them a soil sample so that they can do a genomic test on that soil and find something that maybe is different, or maybe it's like the new cannabis fusarium that we've all been looking for, but it's all about having the most number of samples available and in the database.
So I think this is this goes back to scouting, and one of the most important things you can do is try to be an expert, learn about all the insects out there. Because when I first started a long time ago, you see those two insects there. One of them is a caterpillar and one of them is a hoverfly larvae. And if you kill the wrong one, you're killing the one that's helping you. So which one do you think this image is with the green and yellow? Do you think that's the caterpillar, or is that the hoverfly larvae? Right?
And let's actually wait for some chat responses.
Yeah, I'm looking at the chat right now. So I mean, that's I guess to expound on that a little bit. You're not just looking at, oh, there you go. I grew some, got it. Left field got it. The mop top got it. Gorski says a snail. That's just impressive. No, it's just a joke. But the most important thing, it's like when scouting, I'm not only looking for pests, I'm also looking for beneficials. I suppose that's actually a huge component that I left out. So I guess I'm glad we're covering it now. But that's the caterpillar. And
that's also a hoverfly.
That is?
Yeah.
Oh, well, see, there you go. See?
Right? I mean, Lauren, I just want to I want to like, be the sole arbitrator.
No, that looks like a caterpillar to me.
That looks like a hoverfly larva to me with the.
Is it because of the tapered? It's the tapered.
Now that looks like a hoverfly for sure.
Yeah, and this is a great example. Here you go. We have, 3 people involved with IPM, and we're still trying to figure out from a photo, what it is. Now, with the hoverfly, what you described to me, Matthew, is that it tapers. Obviously, it's not a great photo, but if you go back to the other photo, Peter, did you feel like that?
To me, I feel like the tapering head is like
on the upper side of it.
Yeah, it's not very obvious in this one. The reason why I say it is the markings on the back. And, I mean, but like this is actually a better reaction that that there's some confusion, because I think that speaks to the nature of, like it was. I thought this was planned. I thought this was a trick question actually. But like, so you see those on the sides of the specimen, there's these little like poking, almost like spines. And I associate that a lot with the hoverfly larvae, and also those dorsal markings are very hoverfly-esque. I have observational footage on my channel that I like to use for reference, but they can be green, they can be brownish color, they can be kind of an orange color. I would really be able to tell if it was a moving image. If it was a video, I could probably tell better. But
well, there you go.
I mean, that's just. I don't want to like take away too much from this, but I think that's actually a great example of how difficult it can be to tell things visually.
There you go. That's exactly correct. That's how difficult it can be to tell things visually. Precisely.
I have a point about lady beetles. I have 2 points. The first point is that I think that in my experience, I don't know about you all here, and I'm curious what your impressions are in your guys' experience, but basically, I know a lot of people who like to use lady beetles, like the aphid feeding lady beetles, and they think that they're going to eat things like thrips, like whiteflies, like spider mites, things like that. But in fact, there are specific commercially available species that will go after things like mealybugs, whitefly, and spider mites. There's a spider mite lady beetle, a whitefly lady beetle, and a mealybug lady beetle, to name a couple or a few. Has it also been your experience that people sometimes use lady beetles as a catch-all, kind of? I do
think that that's the case, but I also think that ladybugs do the best on some things like aphids. I find that they are the most effective when treating aphids. But then, to like what you're saying, Matthew, there are other targeted species, predator mites like Amblicius swirsky, Amblicius andersoni, or Amblicius californicus, which are all predator mites that are better suited to go after spider mites, broad mites, or russet mites. But another thing to keep in mind with ladybugs is that they're widely available at your local garden store, which is great. They're all harvested from the Sierra Nevadas during the breeding period of that species of ladybug. So they're not a lot of different varieties. I won't say all, but a lot of garden store ladybugs are harvested directly from the Sierra Nevadas and aren't bred within a facility like a lot of our other predator insects. And that's a problem, obviously, for native populations.
Wildcraft.
This is a great point. We might as well tangent
also
into
going
that
to Axoletus. The Harlequin lady beetle. That's an exotic invasive. It even eats native lady beetles, for those who don't know. And it also it'll predate on natives. It'll transmit microsporidia, I think, and other like pathogens to natives and outcompete them. And people will not know this, and they'll buy these really cheap lady beetles and apply them. And I think that's also one facet of biocontrol that it's very important to me, is that people are aware of the products that they're getting, that the organisms that they're using aren't going to be too broad spectrum or have some sort of ecological issue. Russell brought up Bouveria bassiana earlier, and so did you, Lauren, and I'm a huge advocate. People who follow me know this. But it's also a broad spectrum entomopathogen. And so, sometimes I wonder about the sort of the ecological harm of applying some of these broad spectrum biocontrols. Although I do know Bouveria is in a lot of places already naturally. Unlike us, we have an adaptive immune system. We've got like white blood cells and things. We have a personal army inside of us that we create. plants contract out. Plants get mercenary microbes around them, in them, to sort of do a lot of this work. And like I said earlier, sometimes plants escape that environment where they need that mutualism to like be more fit. But then when they escape it, they lose those genes, they lose those. So that naturally happens a lot of the time. At the same time, those symbioses can be very difficult or impossible to gain back. It might take a long time, it might take the evolution of, another pathway that's unique again. So I think I agree with you, Russell. I think that when we look at these, like there was a group that took out a gene cassette in cannabis in order to not allowed to produce THC. Well, unfortunately, in that gene cassette are fungal resistance genes for like Fusarium, Botrytis, powdery mildew. And I think Kevin McKernan of Medicinal Genomics, was talking about that. And, that's an unintended consequence, but like, I think it's sort of abhorrent to tell people that they have to apply, literally have to grow a plant that like lacks these resistance genes when we already are kind of lacking in them perhaps, or could use a little bit more help. I just think that it's sort of unfortunate.
I think so. I think it's sort of like terrain. It's like Antoine Beauchamp's terrain theory. It's like the plant has the capacity to do a lot of the work, but all the other variables have to be right. You have to have the right soil microbiology, the right nutrient levels, the right environment, and then you don't need to deal with the other stuff. But that's a bit oversimplified, especially
I think that it's. Sorry, didn't mean to cut you off, Russell.
Well, that's fine. I was done. Cool.
I do think that it's important to understand too that while you may be breeding resistance to one thing like powdery mildew, that one strain may not be resistant to botrytis or may not be resistant to Fusarium, or we could get a Fusarium resistant strain that's not resistant to powdery mildew. And that's kind of that's the stepwise development of these cultivars, is understanding that you may be able to develop resistance to one thing but not to another. And so when you're deciding what cultivar, what strain to grow at your farm or your operation, understanding what kind of what diseases are you're at biggest risk for. If you have a crop that's going to be at high risk for getting powdery mildew, choosing a powdery mildew resistant crop is going to be best. But if you have a place that's experienced Fusarium before, maybe you want to give up that powdery mildew resistance for Fusarium resistance. But again, I mean, these are all like future developments.
Talking about resistance, immunity, which have very specific, meanings. We get to this point of like, how do you even quantify it? One thing that burns me up sometimes is that people, who breed cannabis in particular, but other plants will say, oh, my plant's resistant. I know because it just doesn't get powdery mildew, or I know because, these plants over here had powdery mildew, but this one didn't over here. And that's not really good enough. That's not really good enough quantification of resistance and immunity, because especially at least from a genetic component, like we were just talking about, sometimes genes are specific or groups of genes are specific. Sometimes it's general to fungi. Sometimes it's a specific fungus, sometimes it's a specific species. So as we go forward, I'm curious to see people actually able to like quantify it. If I'm going to pay a premium, or if I'm going to have as part of my IPM plan for a client, if somebody's trying to grow powdery mildew resistant cannabis, I need them to be able to prove it.
Right. And I think it is a good point to make that there are different species of powdery mildew. And especially if we're talking to a national audience, you may not be seeing the same powdery mildew on the East Coast or in the Midwest that we're seeing on the West Coast. So not all, like a Bacillus subtilis or a Bacillus pumilus may not be the appropriate fungicide to use for your specific strain of powdery mildew.
All right. How about this guy? This guy looks like he causes damage to your buds, right?
It doesn't look like a budworm moth to me. Mostly those are Helicoverpa. This looks like maybe a Maybe a crambid, maybe a snout moth or something like that. I'm not sure.
But in terms of which, like for example, like I see the white, like look, there's the white one.
That looks like a cabbage butterfly to me, potentially. Or maybe a sulfur butterfly that's yellow and not white.
Hold on, Russell, you're muted. All right, now you're unmuted. Now drop knowledge.
Well, for, treading lightly here, it's like Matthew said, there's ones that look like similar to that. Like you called it a sulfur butterfly, but it's a cabbage. It's the one that's similar to that, where it's the cabbage butterfly. Those are the ones that we see have those caterpillars in our area that are causing the damage. It's the little white ones that flit and float around everywhere. That, at least where I'm at, that those are the ones. They look so pretty. Oh, they're so pretty. I go out with my kids. I pay the kids with nets to go catch them. Like 25 cents a butterfly, because it's worth it. Yeah. Those little bastards.
Well, it really does depend on how many you have. A lot of times budworms aren't a problem every year, but being able to tell when you have those issues. You shouldn't just be spraying preventatively for budworms.
The most recent question was from Potent Ponic Steve. What other new bugs are you all seeing? Lace bugs being ones that come to mind. What's the new?
I've seen a lot of lace bugs lately. Somebody recently contacted me. You know what? It was they were getting bordered plant bugs. And also I see a lot. I've been seeing more and more examples of actually ants. Not always am I able to tell the species. I'm no mere mycologist, but ants actually going after the bark or the cambium layer of cannabis, and also termites for that matter, like literally stripping off the cambium. And I think in some cases they're fire ants, but I actually haven't been able to ID them totally.
So I don't know. Have you connected the ant presence to a pest that produces honeydew?
No.
Okay,
no.
Because that's something that's well known, and I'm sure you guys already know this, but I'll say it for the people in the chat who may not. Ants will actively farm insects that produce honeydew for that honeydew as a food source. So that's things like whiteflies, mealybugs, scale insects, those kinds of things. Ants may intentionally farm those insects. So if you see ants, it could be a sign that you have an infestation of something worse.
That's a great
point. I was working with somebody who had Cremitogaster ants, and those are known, for example, to actually harvest from rice root aphids, root aphids like Pemphigus, but not necessarily, I think, rice root aphids. But yeah, in this case, they didn't seem to be, and I wasn't on site, but they didn't seem to be like, having a relationship. There was no trophobiosis.